All right, so in this video, we're going to be talking about the cell cycle as well as mitosis. So what you're looking at here is this is the cell cycle. So you're going to see that there's a bunch of different boxes and there are some stars with a bunch of letters. But let's go back for just a second. What is a cycle? A cycle is something that repeats itself, right? So after something cycles, it's going to do that same process again. So it's repetitive. And so if you're looking at this carefully, you're going to see some letters. So I'm going to start on the right-hand side with the yellow arrow. This is known as G1, okay? And G1 is going to be the longest phase of the cell cycle because it's doing its normal cell function, right? It's growing, it's synthesizing proteins, and it's just doing what it needs to do depending on where it's found inside the body or inside the organism, wherever it might be located. And so then we reach this point called G1 checkpoint. Okay, we're just going to make sure that the cell is healthy here. We're going to make sure that it's got the proper nutrients, as well as organelles working properly, and so on and so forth. And once we reach that checkpoint and everything ends up working out okay, we're going to jump into the S phase. Okay, and in the S phase, this is where DNA replication happens. So if you remember, DNA, it looks like a double helix. There's going to be a picture of it coming up here soon. It's actually going to duplicate itself. So this is also known as DNA duplication. We're doubling our amount of information inside the cell here. Okay, so that happens in the S phase. And once we're done with the S phase, we're going to jump into G2. And all that we really do in G2 is we're just preparing for division. Okay, we're just making sure that we have duplicated DNA that happened in the S phase. Okay, and we're making sure that we're ready to divide and split into two new daughter cells. And so at the checkpoint, we're just making sure, hey, did everything work out okay in DNA replication? Are our organelles doubled? And so on and so forth. Then we jump into mitosis. This is known as regulated cell division. So the cell is going to officially split here, and we're going to see two of the exact same cell as what we started with at the beginning of G1. And so what you see at the M checkpoint is we're just making sure that the chromosomes split accordingly. Again, we're going to talk about that later. There's going to be some good pictures for you to see, and then so on and so forth. The letter C here represents cytokinesis, and this is going to be the official split. And we're going to split, if we're in an animal cell, we're going to split right at that cleavage furrow, which is the pinching of the two cells. We're going to divide the cytoplasm into two brand new cells. And so what do those two new cells do? They jump right into G1, and they go through this process over and over and over and over again. So as you were looking at the diagram in your note outline, there might be some questions. And so question number 1 says, what is the cell checking for at the G1 checkpoint? It's making sure that it's got the right size, the right shape, as well as the proper nutrients. Question number 2 says, what gets copied during the S phase? We said that that's DNA. Question number 3, what is the cell getting ready to do? It is going to divide through what is known as mitosis. And then lastly, what is the cell checking for at the G2 checkpoint? We're just making sure that we have accuracy in that DNA replication. Did any base pairs get swapped out? Are there covalent bonds between our phosphate and our pentose sugars? We're just making sure everything is lined up here. So mitosis, we said that it is the process by which a cell replicates its chromosomes and then separates them into two new identical daughter cells. So this cell, it says down here on the bottom right of the diagram, it says that it contains 46 chromosomes. At the very end, you'll see that there are two new cells, but yet there are still 46 chromosomes each. And in this diagram, they don't have 4 chromosomes, they only have 4. But you can see that there are 4 different colored chromosomes. So there's a light blue, maybe an orange, a green, and then a dark blue. And you can see that they separate, but there's equal information going both ways. So there's 4 chromosomes here and there's 4 chromosomes here. We're going to talk about chromosomes a little bit later. But what is being divided? It's DNA in the form of chromosomes. And on the next slide, we'll kind of unpack what that means. And then what is the cell checking for at the M checkpoint? We're just making sure, hey, did the proper amount of information go each direction? So if you're following my cursor in anaphase, there's a bottom half right here, and there's a blue, an orange, a green, and a dark blue. We need the exact same going the other way. Then there's cytokinesis. We said that this is The formation of the cleavage furrow, I mentioned that a little bit earlier, but it's that pinching. So it's like if I were to take my two fingers and pinch in the middle of those cells, you could see that that tells the cell, hey, this is where we're going to divide. And so what is being divided here? It's those two identical daughter cells. So in this picture, you can see there's 3 red chromosomes on the left, there's 3 red chromosomes on the right. So we're cutting those two cells apart right down that middle. So interphase. Interphase includes G1S and G2. So there might be a question that says something like, which phase takes the longest in the cell cycle? Your answer should not be G1S or G2. It should be interphase, because interphase includes G1S and G2. Okay? So then we got mitosis over here as well as cytokinesis. But what we're going to do is we're just going to unpack very quickly, once again, what happens in G1S and G2. So in G1, the cell, right, it's growing, it's got those normal functions, and it's preparing for what is known as the S phase in DNA replication. So the S phase, like we said, is DNA replication, right? We're copying that DNA in the S phase. And then once we're done with the S phase, we're going to jump into G2. This is where normal cell growth is going to occur. We're going to duplicate our organelles here, and that's important because we need all cells that are going to be produced to make sure that they have the proper organelles. We're also going to synthesize some proteins here, so we're going to make some proteins, and then we're also going to check for errors in DNA. So just making sure that, again, no base pairs were substituted for, and everything lines up accordingly. You might be looking at the bottom right down here. There is a diagram that shows you a little bit more of the cell cycle, but you'll notice that on the very outside, there's this letter I that's in orange. That indicates interphase. This isn't necessarily drawn to scale, but it shows you that all 3 of these, G1, and G2, are located in interphase. There's also a G0. You might be wondering, what the heck is that? Well, after G1, G0 is what is known as a resting phase. And so, as you may have heard in the past, your brain cells do not divide very often. So if somebody says, oh, you may lose brain cells because of this, well, there's actually some truth to that, because brain cells do not go through mitosis. So you have a limited supply of them. This is where I'm going to stop for today after I explain this slide. But you might be wondering, what the heck is this thing called a chromosome? Okay, so what we're going to do is we're going to start over here and kind of work our way back all the way through into the middle where the chromosome is located. But if you look on the right-hand side, there's this thing called deoxyribonucleic acid, DNA. Okay, hopefully you've heard of this before, but this is that spiraled staircase that we talk about, and it's what makes you and I super unique. Okay, and as we coil it, as it gets a little bit more condensed, we're actually going to wrap it around these things called histones. Okay, and histones are proteins. So when DNA wraps itself around histones, it eventually becomes a nucleosome. Okay, so all of these pieces that you see here, these little discs, okay, these are nucleosomes. And as you can see, the nucleosomes they condense together and they form a shape that kind of looks like a tube. And as that tube kind of continues to wrap itself around each other, that actually becomes a chromosome. So chromosomes actually contain histone proteins, but DNA is just wrapped around it. So chromosomes are the most condensed form of DNA that you can find. And so as we continue to go through this unit, we're going to dive into the stages of mitosis using language that aligns with chromosomes, chromatin, and DNA.